I'm so appreciative of the invitation and the opportunity to speak to the community. And I'm excited about these results. These are kind of earlier results. This is not the product of decades of research. So I'm eager to get questions from the audience today. And yeah, so we'll go ahead and get started here. So before I get into the talk, actually, I just wanted to have a few things up front. So first of all, obviously there are different opinions about language choice when talking about autism. And I just want to say that both person first and identity first language both have valid arguments. And I tend to use a mix because sometimes depending on the context, one seems more appropriate than the other. The other thing that I'll say is obviously today I'm going to be talking a lot about language. And I think one of the things that you could say about this, well, one thing that I want to make very clear is that not everybody has words and not everybody communicates in the way that the majority of the general population communicates. And I want to acknowledge that spoken and written language are not the only forms of communication. But what I'm going to be talking about today is pilot research. And we're trying to test the feasibility of certain ways to assess language ability in a large number of people and to do it in an automated way, in a way that doesn't require a lot of one-on-one interaction, because that would be prohibitive for our purposes. And so because of that, this first pilot study is focusing on independent adults with and without autism. And so obviously that skews things to people who have that level of language ability. And so all of the research that I'm going to present today is colored by that. And so I acknowledge that as a limitation. The other thing I want to say is that I am a computational geneticist and I'm not a clinician. I'm a Ph.D. scientist. I work with a lot of clinicians, and so the clinical concerns are close to our thinking about research. But I just want to emphasize I'm not a clinician, and I'm not going to have the expertise and background to answer perhaps many of the questions that you might have about language. I'm also not a linguist, and yeah, so I'll be able to answer from my expertise, but there will probably be a lot of questions that you'll have that I won't either have data or the expertise to answer. I think it's also important to talk a little bit about community involvement. So I'm involved with a group that runs a website called dldandme.org, which is specific to developmental language disorders that include many folks with autism. It's also related to specific language impairment, or SLI, which is a term that's not used so much anymore. But I wanted to emphasize that we're seeking others who are affected by a DLD to form a community advisory council for language-oriented research in the context of autism. So if you have interest in that, please reach out to me. I want to frame things today because the talk today was enabled by the Spark community, and a lot of the data comes from the Spark community. But we're interested in language in general because I'm in a department of psychiatry, and psychiatry is unique among the fields of medicine because it's one of the only fields of medicine that can't rely on objective, for instance, blood tests and lots of other really objective readouts. We have to somehow distill down the interaction that happens between a client and a psychiatrist to make decisions about care and assessment. And so here I've got this image where you've got a client sitting there and they're talking, they're saying things, and a lot of the things that they're expressing, both verbally and non-verbally, are being projected out into the ether. And some of those things represent what we call in science traits. And these are things that are durable. So things like your level of cognitive ability would be a trait because that's fairly stable over time. And then in the mix are a lot of things that are state, which are things that are dynamic and that might change over time. So for instance, mood is something that is a state because it can change. And so with all of that, a trained psychiatrist builds an impression, but ultimately there's a lot of subjectivity in that impression too, even if they use the best tools that are available. And so one of the things that we're working on is how to make the data that are inherently there a little bit more objective so that we can more readily use them in research. And so this is a little bit of what it looks like when we're trying to quantify that intuition that a psychiatrist might have. That we take recordings that happen in various contexts, and I'll show you that in just a second. We can extract things like acoustic features, so the sound of a person's voice, and there are all sorts of different qualities of voice that actually carry a lot of information. A lot of research, for instance, in Parkinson's disease, focuses on acoustic attributes of the voice because there are inherent motor features embedded within that. And then there are the semantic features, or about the meaning of what a person says. And so we use machine learning to build objective and quantitative mental health biomarkers that are based on speech, ultimately. So just a few examples of how we collect speech. We have a language battery that I'm going to talk a little bit more about called Lingo. And we get good coverage of a variety of language domains in about 30 minutes. And then we have also piloted a soundproof booth that has a tablet mounted inside it. This is placed in one of our outpatient clinics, and people can spend 3 to 5 minutes in there completing a simple language task, and then they donate that data to us so that we can analyze it and do our research. And then we also have developed what we've called the Iowa speech sample, which is a fairly short task that can be completed that elicits speech, and that we hope will eventually be kind of like a vital sign that is taken routinely at clinical appointments over time. And so this is Hancher Auditorium, which is the fine arts or performing arts center at the University of Iowa, and it holds, depending on how you look at it, 1,500 to about 2,000 people. And that's about the number of people that participated from Spark in the study that I'm going to talk about today. For the sake of time, I'm going to kind of skip over this, but I hope I've made the case that speech and language are really important reservoirs because they're linked to everyday outcomes, but they're also really cheap to collect, and we can there's a lot of discovery that we can do from a scientific perspective. So today I'm going to give you a tour of some of our initial findings and do a few deeper dives on some of the specific tasks and domains. And again, just to emphasize that this is very new, so there may be more questions than answers. I'm also going to just say a quick word. I'm going to show a lot of examples that come actually from the data that we collected. And to preserve people's identity, we've manipulated the sound files a little bit to obscure the identity of these people while still trying to make it sound natural. So in case anybody's wondering about that. So again, language traits are genetic in that language traits depend on brain development and brain function. And we know that brain development and brain function are substantially genetic. And so this is sort of the path that we envision as we think about the relationship between genes and language, and then the ultimate outcomes that individual differences in language are related to. The other thing I'll say is I use language in a very hand-wavy sense, but there's a lot more precision that goes on when we think about different aspects of language. So if you imagine light passing through a prism and getting separated into its constituent components, you could similarly think about how language is actually made up of a lot of different domains. And this is by no means exhaustive what I've listed here, but just a few examples of different aspects of language, different dimensions of language. This was said in sort of the summary of the talk for today, but I think it's important that we continue to build a scientific understanding of individual differences in language ability. And one of the things that will ultimately help is in developing a more inclusive community, understanding those differences and developing the means that we need so that communication can happen across the divide of those differences. Some of these factors that influence individual differences are innate and biological, like genetic variations, and some are influenced by the environment, like the environment that was provided during language acquisition, for instance. So right now, language assessments are typically done in an in-person context. So here I'm using the Self-5 as an example, where you would have a trained clinician sit down with somebody. There's a flip book, and there are other materials that are used to assess language. Unfortunately, this isn't really a scalable option for genetic research where you need thousands of people. And some of the other limitations are that this is really meant for children and adolescents. It's not meant really for adults. There's a low ceiling, and it requires, like I said, a proctor. Can't really be done online. Still, it's a validated instrument. So as we were developing our own tools, we kept an eye on whether or not the outcomes correlated with what was delivered by the Cell-5. So now to the tool that we developed. So like I mentioned, this is called Lingo. It's an online tool. You do it through your browser. And in about 30 minutes, there are a variety of tasks that cover things like fluency and vocabulary. We get estimates of nonverbal reasoning, of rhythm entrainment, and expressive language ability through narrating a picture. I'm going to talk about a few of these today. So this runs in the cloud on Amazon Web Services, and it uses the microphone. We don't record any video, we just record audio, but it uses the webcam's microphone to record that audio. And this is totally unsupervised. And so that's both a good thing and a bad thing. The chickens come home to roost a little bit when you have to go to extensive lengths to QC the data that come out of this, because of course there are people who are cheating, or there are other people in the room, there's a TV on the background, and other things that we ask people not to do still happen. So we have to get means to detect when that happens. So just a few examples of what this actually looks like. Here's a screenshot of the actual assessment. This is one task where a person's given 30 seconds to name as many words as they can. We record the audio and we transcribe it using online APIs or online tools provided by companies like Google and Amazon to process that data. And then we extract various measures out of that I'll get a little bit more into later. Here's another task where we ask the participants to read as quickly as they can from left to right and top to bottom. And then we get that waveform, we check it for various things, and then we can pull out certain scores, certain features that are inherent in how they performed on that task. So we performed a research match in which we recruited about 1,500 independent adults from Spark. This took about, so a little over half of those individuals are autistic. This took about 6 months or so, and now we're recruiting a second wave for an additional 1,000 people. And we also asked at random 200 people to take it again 2 weeks later so that we could look at test-retest reliability. And so as expected, our analyses support the various aspects of language that I mentioned before. And the nice thing is that these participants are already genotyped or sequenced, and we're finding genetic associations in there. So the first task I'm going to tell you a little bit about today is a sentence repetition task. So this is seen by many as the single best task that you can do to get a read on a person's overall language ability. So there's a really strong verbal working memory component, and this will become clear when I tell you what this is. So the participant hears a prompt sentence, and these are of varying difficulty. And then they have to, there's a brief pause of a couple of seconds, and then they have to repeat it back while they're being recorded. So for instance, an easier sentence would be, didn't the boys eat the candy? There'd be a pause of a few seconds, and then the person has to repeat that back. And then this progresses up to a very difficult point. And we purposely made these challenging. I essentially have heard these hundreds of times, and they're still difficult for me. So I hope nobody feels bad when they when they're presented with these, because we purposely want to kind of break people a little bit so that we can see where you break. So this sentence is, having been tardy more days than not, the brothers knew it was only a matter of time before each would have to be responsible for himself. So most of the time people have struggled with that quite a bit. There are a few people amazingly who can do that, but they are rare. So the way that we score this is kind of outdated text. It's we use a different speech to text algorithm now, but we translate that, transcribe it, and then we split the response into bigrams, which are simply pairs of words as you kind of slide across the sentence. And then we count the number of matches to the actual prompt sentence. And so that can give us kind of a quantitative score, which is nice because actually this task has been done in a lot of other contexts, but usually you only report whether or not the person got it right. But here we can report the degree to which the person got it correct, which is useful. It gives us more granularity. So what we did is we took people's performance on this task as measured by, how many they got correct. And we scanned through the genome here. I'm just showing some chromosomes, and we were looking in this case for rare variants, rare genetic variants that were damaging, that could explain, a clear departure in this task, which is largely verbal working memory. And so here's one of the things that we found. There's a gene called PDE8B that we found to be significantly associated with performance on this task. And so you're seeing that on the y-axis here. So the higher you are, the better you are performing on this, and the lower you are, the worse you're performing. And we found that these people over here carry rare and damaging variants in this gene. And so we found that they were significantly more likely to perform worse on this task. So this gene's what's called a phosphodiesterase, and all that is a protein that is involved in key cellular communication roles. And it turns out that this gene has actually been implicated in other case studies in what they called autosomal dominant striatal degeneration. And those case studies reported signs of Parkinsonism, including dysarthria. And so I went back I saw that and I was kind of interested. And so I went back and listened to some of these, expecting to see signs of dysarthria, which is weak or slow or slurred speech that's difficult to understand. But as you'll see here, so this is starting with one of the easier sentences, and this is what the prompt sounds like.
The rabbit was not put in the cage.
Okay, so that's the prompt sentence. And then I was listening to, so these are two different carriers.
The rabbit was not put in the cage.
The rabbit was not put in the cage.
Okay, so I didn't hear any sign. Nothing of that sounded like dysarthria.
The rabbit was not put in the cage.
All right, it looks like that got stuck in there one more time.
The rabbit was not put in the cage.
The rabbit was not put in the cage.
Sorry about that. But I continue to listen to these recordings of their attempts to repeat the more complex sentences that occur later in the task. And this is where the PDE8B carriers seem to have something in common. So
the cafeteria doors, around which the 7th graders sometimes stand until the lunch bell rings, are always closed, but never locked.
Okay, so that was a very difficult sentence to repeat, but listen to the response.
Can't repeat it either. I can't remember.
Blanking. The doors, something, are never locked. My bad.
Okay, so most people are not going to get this. They're not going to nail it. But what is uncommon, most people will get out a little bit, and especially something that sounds kind of like what the prompt sentence was. But what was different here is that they really, really struggled a lot more than most people do.
The cafeteria doors, around which the 7th graders...
remember.
Sorry about that. Seems not to
Can't
go to the
repeat it.
next. There we go. So this is the prompt sentence right here on the x-axis, and then on the y-axis, their accuracy. And so you can see over time the carriers of this gene variant are in red, and they are shifted downward, meaning that their accuracy is actually lower overall. And then by the final sentences, many of them just give up and they can't get anything out. So this was really striking, I thought. And so I looked at, we have a school age cohort. They're not autistic, but we also have genetic data on them. And this looked to see if we could find anything there. We couldn't find anything. And like I said, the literature indicates that this is involved in degeneration, right? So that got me wondering whether age is a necessary ingredient for this gene to exert an effect on language. And so what we found when we looked at age, So in the carriers are in red here. And so you have age across the x-axis here, and then their performance on this verbal working memory task is here on the y-axis. And so even though there is a main effect, there's this interaction with age that if you're in your 20s and you're a carrier, you're basically in the normal range on this task. But as you progress from that, there is this downward trend in performance. And so it suggests that what we're seeing really is like a degenerative trait here that's picked up by this verbal working memory task. So again, this is a gene that's expressed in a part of the brain called the striatum. And this is also a part of the brain that's known to be involved in verbal working memory and information gating and sequencing. And so it makes sense that people who have this genetic variant are not going to perform as well on the sentence repetition task. But again, I want to point out that one of the things that we did here was we raised the ceiling a lot to the point of breaking, because if we had kept that low ceiling that exists on a lot of these clinical tests, we would have missed this. We needed those more difficult sentences to really show just how hard this was for these people. So I'm going to move on to another task here, a verbal fluency task. And what this looks like is we give participants these letters, and then we ask them to just name as many words as possible to begin with that prompt letter. We give them 30 seconds. A lot of other variants of this task give a full minute, but we actually find that most people kind of peter out by 30 seconds anyway. So for time efficiency, we do 30 seconds. So, and traditionally, people are mostly interested in the number of unique words that you name, and we are too. That's an indicator of verbal fluency. So if you've got 30 seconds and you're just rattling off words, and it's not difficult to you, that is an estimator or an indicator of your verbal fluency. But there's a lot more going on than just that. And so one of the things we can do is look at vocabulary size, and we estimate that by looking at the words that you say, how rare are each of those in what's called a corpus. And this is basically a huge assembly of text. You can imagine Wikipedia is often named as a corpus of language or of written language. And so we would look to see how frequent these words are in that. If they're very rare, then that suggests you have a very expansive vocabulary. The other thing is that we can build a multi-dimensional semantic space, and hopefully that'll become a little bit clearer in the next slides I'm going to show you, based on all of based on the corpus of all of the words that people said in this task. And people sample this space in different ways that I'll show you in a few slides. But first, I'm going to give you just a couple of examples so that you can kind of develop an intuition for how you might think about estimating vocabulary size from this. So here's the first person.
A. And, ask, am, add, are, ate, art, ant, anthill,
Okay, and that was it for that one.
apple, apex, agape, aghast, and apricot, artichoke, asparagus.
Okay, so the thing I want you to note there is that vocabulary, size of your vocabulary and your fluency are not the same thing. And we've sort of seen that those kind of can be disentangled from each other. Oh no, I did it again.
A, apple, apex.
You gotta remember it doesn't advance when I do that. Okay. So again, so you can just count the number of words. You can check vocabulary, but we were thinking more deeply about this because if you listen to enough of these, you start to wonder what is going on in people's minds, and is that meaningful? How people sample the space of their own vocabulary. And so we were thinking about whether there might be risk words. And what I mean by that is, are there words in this task, or are those words uttered with equal probability? Is it just basically your brain is like a giant bag and you're reaching in there and just randomly grabbing stuff? Or is there potentially a deeper meaning? So we already demonstrated that you could look at vocabulary size, but beyond that, are there certain words more immediately within reach for certain kinds of brains? So for instance, are there schizophrenia words? Are there depression words? Are there bipolar words, et cetera, right? So obviously we would not expect any of these words individually to really be very predictive at all. They would have a very weak effect. But if we take the aggregate effect across all these different letters and trials, can we start to build up, yeah, this person's kind of saying these words, which is not a common thing to do. And so that's kind of how we're thinking about that. And especially, do these relate in any way to latent genetic risk, and even in an undiagnosed person? So one of the things that we looked at sort of along these lines is parts of speech, because we didn't give any instruction on the parts of speech that people should stick to. And so we got everything. And as you might expect, this is the proportion of all of the words that you say, and then they're part of speech. And as you can see, it's most common for people to say nouns. Nouns are the things that are most immediately within reach for people, and to a much lesser extent, verbs, adverbs, and adjectives. But if you correlate this with some other variable of interest, and here we were interested in bipolar disorder, which I'll get to in just a second. So this is polygenic risk for bipolar disorder. This doesn't mean you're diagnosed with bipolar disorder. It simply means that on average, you have a little bit more genetic risk for more or less genetic risk for bipolar than the average person. And so we found that as you increase a person's genetic risk for bipolar disorder, the proportion of nouns that they name goes down, and the proportion of adjectives that they name goes up. So there's something about genetic risk influencing brain development, brain function in a way that makes it statistically more likely that a person is going to reach for descriptive adjectives when they're performing this task than nouns that are just enumerating things. So again, thinking about this spontaneous accessibility, and in some way, it's strangely genetically influenced. So there's preferential access to descriptive words, and this may underlie the overrepresentation of individuals with bipolar disorder in the creative arts, and particularly in writing. So there's been some other research that's been done on this. We've also done research on this and found that there's a pretty robust connection between genetic risk for bipolar disorder and propensity, especially for writing and especially for poetry. Yeah, so this is in agreement with other data that we found is very interesting, and we'd like to get to the bottom of it. And so I mentioned a semantic space. So you can organize, if you took the giant bag of words of all the words that people said in this task, 1,500 people and all the words they said, and you can kind of actually map out, you can arrange things so that words that are semantically similar to each other, or they're thematically related to each other, are closer on this map, and words that are more different are farther apart. There are algorithms you can use to do this. And so we did that and organized into 20 different topics. And you can see kind of highlighted in red, it looks kind of like a conch shell here. Since this is a PG talk, I blocked out profanity, but as you might imagine, profanity is one of the categories that comes out of that. And so the fascinating thing about this is that the semantic space is sampled in ways that correlate with age, that correlate with your sex, that correlate with whether or not you have a diagnosis, and that correlate with genetic risk. A lot of those are related to exposures and society, right? So we found that males were much more likely to say words that were related to mechanical or automotive or sports related kinds of things, which is very stereotypical. And females, again, stereotypically were more likely to say words related to clothing and food and other people. Right. So it sort of confirms some of these stereotypes that are partly biological, but also partly environmentally influenced. But we observed those. And so that gave us confidence that there really is something to this. And so, yeah, the questions here are, do people traverse this? We're calling it a solution space, but you can imagine it's like the complement of all of the words that you know and what comes most naturally to you when you're put under pressure. So do people sample or traverse that space in the same way? And if not, what do those differences mean? Are the differences in the hardware, like the brain structure or shape or connectivity, or is it software only, like the upbringing that you had or cultural exposures that you had? So exposures could be things like education, profession, hobbies, things like that. There might also be some more innate differences that actually might be related to these exposures that you seek them out. And so we're very interested in, this is ongoing work, to think about whether word sampling differences provide a kind of brain map. So I'm going to show you in the next two slides, two examples, and I want you to think about what's different between these two individuals.
Cataclysmic, charismatic, charisma, comorbid, cosmos, cantankerous, creatine, Coptic, champion.
Okay, so that's the last one for that.
Cameron, cat, Charlie, couch, cook, chef, chair, charge, change, carrots, cookies, cereal, car, coffee.
Okay, so I hope that you can appreciate.
Cameron,
No, come on, there we go. I hope you can appreciate that these two people drew from very different parts of this giant space comprised of all the words that people said in this task, and it's been organized according to thematic or semantic relationships. They sampled this in very different ways, and we constructed this space in a way that tried to optimize words that were more or less associated with genetic risk for certain kinds of conditions. And so this long axis here is sort of associated with genetic risk for bipolar disorder. And it's interesting that again, this sort of confirms what I was saying before, but a lot of the more rare words that are indicators of a more expansive vocabulary are showing up in the region that's a little bit more associated with genetic risk propensity for bipolar disorder. Okay, so that's the verbal fluency task that you can actually extract a lot of other things out of, and we're continuing to do that. The final task I'm going to show you is a picture narration task. So I'll just give you a few examples, and these are not extreme examples, but I think you'll notice that they're very different from each other. So think about, as you listen to these, and again, remembering that the instruction is to describe the scene in as much detail as you can in 30 seconds. I want you to think about I want you to think about a couple of things, but first I want you to think about what factors make one participant have a better expressive language ability than another. And then secondly, if you were to close your eyes and just listen to this narration, would you be able to faithfully visualize the actual scene that is in front of them based only on what you're hearing from the person describe?
There are people sitting on a long sofa. The sofa seats 3 to 4 people, but there are 3 people sitting on it. One person to the right of it has his feet up and is smiling. Looks like he's having fun. He's wearing a black shirt, black hair, brown skin, tan pants. The man in the middle is leaning with.
Okay, so that's 30 seconds, we're up there, and then listen to the second one.
Okay, so there's 4 people on the couch. I think they're friends. The couch is brown. There's some sort of bag on the table. The wall behind them is bare concrete. They seem to be pretty relaxed. Two of them are smiling. The other two are kind of just looking at other stuff. There's some bags of, I think, flour, maybe coffee on the shelf behind them. They're probably roommates. There's an air conditioning unit on the wall behind them, plugged in, maybe heat.
Okay, and then that's it. So think about the different details that you got from either, and what was emphasized in the narration in both cases. Oh no, do that again. No, okay, there we go. So here's some example narrations from some of the other figures that we have. So somebody says man, woman, another woman, African-American lady, one sleeping, one alert, blonde hair, white jacket. Somebody else says for this one, it looks like someone parachuting into a city. All you can see is their feet. A mom holding a baby, hugging her other child, comforting her. Friends, brothers on a couch, 4 people, 4 guys, 1 table, a room, a book, something like that. So just a few examples of the narrations. So we had this data, and there wasn't necessarily a book written on exactly how these should be analyzed, because again, everybody doing research on this is sort of interested in different things. But we started to notice as we listened to more of these that obviously the biggest difference is just in how much people say. Some people say very little, and others say a lot. But what we're interested in is what details are communicated. So can a person say less, but still communicate the same general amount of information in some like objectively measurable way? And so an example here of a concise narration that does give you a lot of the specific detail of this image that would allow you to discriminate it from other images is this one up top. Very short, but a lot of the details are there. This rich narration, gets at details like there are 4 guys hanging out. They are in an unfinished basement. They're on a couch. There's an ugly stripe pattern to it. So something about that, they're elaborating. There's a backpack on the table, and they read into that, right? So what does that mean? Maybe it means they're students, probably palling around after classes. So they're kind of projecting, what might be going on here. There's a revision in here. Again, just a lot more detail in that. Again, both of these people were given the same exact 30 seconds to do this. So we looked at one feature, which we ended up not really using, but we were thinking about, well, maybe there's this slow start phenotype, like, because we notice some people, and I even do this when I do this task, I kind of have to get my bearings a little bit. So I'll waffle in the beginning, and the things I'm saying are actually not really helpful in understanding the complete scene. But then I'll recover later, and I'll pick up the rest of the detail and start performing better later on. And so we had a way to kind of come up with this, and basically a high score means that your early words carry little information, but there's a trend of increasing information content. Another feature we looked at is what we call concision, and it's basically how quickly can a participant communicate the most essential details. So our solution for that was to take what we're calling an on-target word score, which I won't go into the details, but we basically trained a machine learning classifier to be able to discriminate between all of the different prompt pictures that we took based only on the narratives that people gave. And so from that, we learned what words were really helpful in discriminating, oh, they're looking at this picture versus, oh, they're looking at this other one. So we could score every word and its ability to be kind of an on-target word. And we just look at the first 5 words. If you have a high score for that, that means you have immediate recognition and communication of what the defining elements of the scene are. And then this richness one, it's actually very closely related to just the number of words that you say, but it's a little bit more than that. We took the sum of the on-target word scores of everything in the narration. So what's the total amount of relevant information communicated? So if you have a high score, then your narration has a lot of unique words that each have high specificity to that prompt scene. And so, as I mentioned, this is pretty strongly correlated with the number of unique words in the narration, but it has a better signal. So these are showing from the consistency across the different prompts, right? So the richness had better Chromebox Alpha, which is a measure of consistency across the different items that you're using. And then we also looked at test-retest reliability. So basically this is trial 1, and then this is trial 2 of the same people 2 weeks later. So every dot here is a person, and they would take it 2 weeks later. And we found that, the people who said a lot of words the first time tended to say a lot of words the second time. And so that was something that was pretty correlated, as was richness, and to a lesser extent concision. And then, like I mentioned, the slow start feature seemed to be pretty variable, and so that was not something that was terribly reliable. So one thing that we found when we looked at case control status of association with an autism diagnosis was that, so here we have autistic people and then neurotypical people. So the autistic people in our sample, and remember, everybody here is a legally independent adult. They gave significantly simpler narrations of the prompt pictures, but we didn't observe any differences in this concision, which is like the immediate detail that's provided. So the essential details seem to be there. It's just that there's not a lot of elaboration of other details in the scene. So we associated these with various polygenic scores. So I'm just looking at the concision and the richness here. We found that people who have higher polygenic propensity for extraversion, which I don't know if you knew that that was something that was partially under genetic control, whether you're an extrovert or introvert, but people who have a genetic propensity to extraversion tend to not be as concise in their narrations, right? So there's more filler there. People who are more introverted by comparison tend to get right to the point in their narration. We didn't see an association with richness when we looked at the polygenic propensity for extraversion. This is for cognitive performance. We didn't see a relationship at all with concision, but we did for richness. So in other words, people who have a higher polygenic propensity for cognitive performance tend to provide more detail in their narrations. And likewise, in the other direction, people who have a higher polygenic propensity for schizophrenia are more likely to have lower richness in their narration. So again, these are just examples of kind of connecting things that we're objectively picking up from people's narrations and then seeing how that relates to genetic propensities for different things. So just summarizing what we've seen here so far, the number of words and richness, which are, again, highly correlated, they're consistent, reliable measures and suggest that that's a trait, right? So that's pretty stable, that's pretty durable. Concision and slow start have lower consistency and reliability and may represent states rather than traits. So things that are more dynamic that might depend on your mood, whether you took your meds that day, other things. And so we found that there were these polygenic associations with introversion, extraversion. I didn't show it, but we also found an association with genetic propensity for ADHD and also reaction time. So we need to do more work on this to derive formulations of these concepts that are more robust. That's one thing that we've learned from this. We also found that autism diagnosis is associated with that lower narrative richness, but not with a lack of concision. So still the essential details are being communicated, but just with less detail. And then the richness is negatively associated with a genetic risk for schizophrenia and positively associated with genetic propensity for higher cognitive performance. So what this does is it raises an interesting question about the possibility that this might be a biomarker for the negative symptoms of schizophrenia potentially. And it's kind of, this is novel because we're sort of imagining a kind of genetic validity here to developing these readouts. Okay, so in just the final couple of minutes before we open it up for Q&A, I have a couple of other things I'd like to talk about. Okay, I hope some of the people on the call right now have had a chance to play around with some of the amazing advancements in AI that have happened over the past couple of years, well, past year really, mostly. One of those is the ability that you can type in some text describing a scene and then an AI can generate an image for you. And one of the things that was quickly learned was that a lot of times the quality of what the AI produces in that scene is highly dependent on the input that you give in your writing, so linguistically. And so much so that there's been a new kind of job, and I actually don't know if this is a real job, but people say that it is, but prompt engineer, right? So in other words, you're a person who is kind of an AI whisperer. You know how to communicate detail to an AI in a way that will deliver the desired results, okay? So I hope some of you have seen this, and maybe you know where I'm going with this, but so here, this is the original prompt, or the original prompt photo in the task. And then here I have two different narrations of that same photo. And so we thought, well, really what we're trying to do is we're trying to estimate the level of, the fidelity that people can communicate what they're seeing through their language. And so what we did is we basically popped these in one of these AI image generators to see what the AI would come up with and whether it looked anything like this. Okay, so here we have 4 guys are relaxing on a couch and using their cell phones. Well, okay, so here's what and don't look too closely at the faces because it's kind of nightmare fuel. But so it produces kind of this generic image of exactly what's being said here. I guess this is the 4th person who's kind of off camera there. Contrast that now with what we fed into the AI here with this much more detailed narrative, right? And so this is what the AI produced. And I think that you can appreciate if you look at this and you look at this one feels a lot more like the original prompt. And so we haven't bottled this really or figured out, we're in the process of kind of figuring out how to more reliably automate this, but we're fascinated by this idea of reconstructing an image purely from a person's narration and then somehow using that as an estimator of the depth and quality of the narration. So in the interest of time, I think I'm going to kind of accelerate here, but so just listing some things that we've noticed go into a rich narration. So one of the last things that we did with the narrations was we wondered whether we could chat with these narrations in the same way that, again, I hope some of you have been able to use ChatGPT, but in the same way that you can chat with ChatGPT, can we chat with the narrations to find things that are interesting? So for instance, can we ask, and this is essentially what we did, where are the people in the narration? Well, this one says, I don't know where these people are. They could be in a classroom or something. What's the weather like? Okay, there's some comment on weather. What will come next? So this is a person who's saying, the last view you see before you fall off the top of a building. Do you feel a personal connection to the scene? It's a very creepy feeling looking at this picture. You feel like those legs are yours, right? So we can actually bottle this up and figure out the extent to which a person's narration answered questions that they didn't even know were necessarily going to be asked. But this way we have different ways to quantify a person's narration. So just some teasers from this, we found that higher cognitive performance genetic propensity is associated with a greater proportion of time describing the non-emotional scene details. And on the other hand, more time spent describing the emotional content is related to more overall polygenic risk for mental health conditions. And then finally, this was crazy, but we found that people who are at greater risk for insomnia, greater genetic risk for insomnia, more likely, significantly more likely to devote attention to the sleepiness of the people in the narrations that they were in the narrations that they were giving. I just want to mention there are some limitations to this work. So, but for the good part, the assessments or interventions that use speech and language as their input, they're going to be cheap. And so there's going to be availability in under-resourced areas. And more generally, text-based AI agents are most likely to have a near-term impact. So think about this. They're cheap, they're immediate, they're anonymous, they're non-judgmental. So huge potential there. But the bad parts are that, whatever bias exists in the model is going to be propagated to anything that uses it. And the quality of the predictions is proportional to the amount of words that we can feed into it. So basically what that means is people with little to no words are unlikely to benefit from a sophisticated linguistic analysis. And finally, that minorities are underrepresented in genetic studies, which is a problem. So the take homes I want you to get from this are that we found that verbal working memory was associated with a variety of mental health issues, and it was strongly related to aggression and externalizing. And we wondered whether that was somehow an expression of frustration. We observed a form of neurodegeneration that was related to this gene PDE8B. We learned that how a person samples their own vocabulary under time pressure is linked to age, sex, diagnostic status, and underlying genetic risk. That autistic people tend to give terser narrations of scenes while still communicating the essential elements. And that some of these features that we pulled out of the narratives correlate with polygenic propensity for things like cognitive performance, extraversion, and schizophrenia. And finally, that new applications of AI will revolutionize using speech as a data stream for research. So I would like to thank especially Lucas Kasten, who did a lot of the analytic work here, but really first and foremost, the Spark team, Casey White and Marty and formerly Brianna, and also Pam, who was very helpful on a lot of these discussions. And then of course, the Spark individuals and families who contributed to this work. So with that, I'm sorry for running, I took 3 minutes longer than I'd hoped to, but I would love to take whatever questions you guys have.
Thank you so much, Dr. Michaelson. I think this was a really interesting way that you presented these findings. It was really, really great to follow along with. So yes, we do have a few minutes for Q&A, so I'm just going to jump right into it just in the interest of time. So this research, primarily dealt with, the spoken language. So whether it was this research or other research that's maybe planned, do you think there could be differences in performance abilities for a similar research, but with writing and written language versus spoken language in these individuals?
Absolutely. I think, again, I think once we get a handle on how to actually do this kind of work in the easiest scenario, I think we should think about other ways to communicate. I am not an expert in sign language or even with the deaf community at all, but I have attended talks that fascinated me on looking at these kinds of developmental language issues in people who sign. Right. And for me, as an outsider, I was like, I'd never even thought about that, but that would translate and you would see many of the same kinds of things. And so I think that a lot of these issues, a lot of these phenomena that we're finding are probably valid across very different kinds and modes of communication. And so I'm looking forward to being able to say more about that as we look at other cohorts.
And do you have any thoughts on, a minimally verbal adult with autism who maybe can't retrieve the large vocabulary that they have in order to speak? So is just any thoughts on that in terms of research?
Yeah, I mean, sort of as I said at the beginning with my disclaimer slide, was that I think that the genetic discoveries that we make here are going to be valid, and they may manifest in different ways for people who don't have words, or have very few words. But the way of measuring these is, of course, I think going to be the biggest difference for how. So it's going to be fundamentally different how you measure it. Yeah.
And so you use the Spark cohort for this research, and so many non-autistic participants are the parents of or relatives of autistic individuals. Do you suspect that had any influence or impact on the results?
Yeah, I think it does. And I think if anything, it often makes our results more conservative than they are in reality, because you have people who are kind of drawing from similar genetic factors. One of them might not be diagnosed and the other is, but that sharing of genetic factors may sort of manifest in the language scores that we're collecting. One thing that I didn't really emphasize is that we found that non-autistic parents of autistic children, their verbal working memory scores were predictive of whether or not their autistic child had a language disorder. right? So you have these people who aren't diagnosed, and so that's sort of indicative of that genetic transmission of factors that are linked with language ability. Now, it may be that the child inherited a disproportionate number of other factors that together made that expression of that trait much more severe. But that was one thing that we observed, even going from non-autistic parents to autistic children.
I had quite a few people in the Q&A that are just interested generally about next steps for this research, if you have any planned next steps.
Yeah, so one of the next steps, we're extremely fascinated with this, which is why we're collecting more data. We're going back in and we hope to be able to collect more variety and more so that we can do a little bit more with the genetic analyses. Now, the other thing that we're adding with this round is that we've sort of realized that even though we are in a position to do much more with this automated phenotyping and being able to do it through the web browser and everything, it still requires some work to do the QC on the data. So it turns out to be not as scalable at the moment as we had hoped. And so one of the things that we're doing right now is to look at how is to develop self-report measures, self-report surveys that we can intersect with these objective measures of language ability, so people can report on their own, like, everyday things that they notice about how they use language and stuff like that, whether you're diagnosed or not, that together can give us insight into strengths in language, but also areas where you might struggle in language. And we're just seeing how reliable is it if people just report these things to us through their own lens compared to what we can measure objectively. That's one of the next steps for us.
Great. And then just a last question from more of the researcher point of view. So the Lingo protocol that was used for this, is that something that is available for outside researchers to use? If not now, would that be something that would be available in the future?
Yep. So we do have a code repository with some instructions on how to use it. That hasn't yet been made public, but we intend, when the paper's published, that we're going to release it to the community so that other people can use this battery for other kinds of research.